All right. Well, first of all, it is an absolutely fascinating topic that one can discover this sixth sense, this magnetic sense that is out there. And I cannot overstate how excited I am that I can still work in this field, that the Virchos have started 50 years ago in a way, and it's still an unsolved problem. Now, the suggestion of why quantum mechanics might matter came from Klaus Schulten in the late 70s. And the basic idea is that you have a light-induced electron transfer, in which case you generate two electron spins that are unpaired, that are now in this transient state, and that then undergo quantum coherent superpositions and transitions from singlet to triplet states back and forth. These are really Rabi oscillations in whatever magnetic field is present, which can be just generated by internal magnetic fields, in particular the hyperfine interactions, so the nuclear spins interacting with the electron spins. But you can also have, let's see how the advancing works. It doesn't. I can't seem to go forward. Let's see. It's okay. Yeah. You can also now, of course, have an external magnetic field to produce an interaction, and it will then change the outcome of this reaction. It can, change the concentration of the products that are generated from singlet or triplet states. Now, this is not only dependent on the intensity of the external field, but if you apply an external field in different directions, you will get different effects. So here is this oscillation that occurs without an external field, and now you can see that if you overlay the external field, you will see this pattern changes. And if you now integrate over what happens over the time course of the reaction, you can see that how much you get through the singlet channel is slightly different depending on which way the external field is acting, and that depends because the hyperfine interactions are anisotropic. Now, what we do, so this is what is the canonical description in a way of the radical pair mechanism that Klaus Schulten proposed. And what is done there is that all of these reaction pathways that lead to destruction of this quantum coherent state, whether it is recombination, whether it is just loss to the ground state, whether it is loss of spin relaxation, spin coherence through spin relaxation, or other processes, are just lumped into a first order kinetics, which works quite well when you're dealing with solutions and so on. And when you do this kinetics matters. It needs to be long enough for these oscillations to actually occur, and therefore give you enough time for these quantum effects to develop. So for a 50 microtesla field, which is the geomagnetic field, you need about a microsecond of spin coherence time. Now, I'm trying to, in this talk, even though it's only 15 minutes, to show also an overview of the field, of the directions that have been done, and potential future directions, as well as hint a little bit of what we've been doing. So one immediate question is, if you had full control over how to build this quantum sensor, what would be good? What would be optimal to detect a magnetic field, and especially the direction of the magnetic field? And remarkably, even this question we haven't answered yet. And in part of it, the problem is it depends on essentially near singularities, on two energy states coming close together, almost becoming degenerate, these so-called avoided level crossings. And if you have multiples of them occurring at an angle where you're interested, then you actually get very strong directional effects. But the smaller this gap is, the bigger the effects are going to be, and it becomes essentially you can't just step through the large parameter space because you're depending on these singularities for effects to come. So even after 20 years of research, you still have certain potential surprises. So Peter Hoare, for example, 2 years pointed out that we thought you just get these sort of generic like sine type patterns of the directional dependence, but you could actually get some real large spikes that he called quantum needles if you have very long spin relaxation times, about 100 microseconds or something like that. Now, one thing, so there's a lot of papers that I can't go over in just looking at the optimality. So that's been a field that's been addressed. What has been addressed much less is the role of noise. So obviously that's the other part in the signal to noise. And what has been studied a little bit is geometric disorder. So obviously for a compass, you would ideally want to have all the molecules perfectly in a crystal ordered. And so people have asked, what happens if it's disordered? And there are multiple groups about 10 years ago that did this. And the conclusion was, by and large, that this doesn't seem to be a fundamental problem. It deteriorates the compass a bit, but even there seem to be multiple ways how you could have some organization that is good enough for the compass to work. There's exactly one study on actually trying to build a signal transduction model and calculating signal-to-noise ratios. That's almost 20 years ago. So that's certainly, I think, an overlooked area in our field. One of the things that we asked is what happens if the quantum parameters change? If you have one hyperfine coupling in one copy of the molecule and a slightly different one in a different one. One of the things you can see, you can build these super exciting patterns if you just think they're all the same. Like here's a quadromodal pattern, which is very intriguing for a compass. But if you now vary these parameters by just a few percent, you see that these patterns change dramatically from copy to copy. So you can't really rely on that. And what's even worse, if you average over all of those, you get a pattern that looks nothing like the one that you started out with. And so the conclusions that you draw from here, you have to be careful when you think about the actual noise. But then there's another theme that occurs in quantum mechanics, and that's that noise may actually not always be negative. So if you look at this quantum needle, if you go to more realistic spin relaxation times, let's say 10 microseconds, this needle turns into a blip. So it's this blue curve here and over here. So that doesn't seem like a very good sensor anymore. But if you now do the same thing that we did before, you start allowing a little bit of variation of the hyperfine couplings, remarkably, instead of going away, this needle actually becomes stronger and it gets bigger. And you can quantify this in different ways and essentially argue that you now can go to regimes of a few microseconds rather than 100 microseconds spin relaxation times, and you still get strong directional effects in there. And we think this is really, it's almost like a band phenomenon that you're getting there, a bit like with the exotons in photosynthesis, that before you relied on energy levels having just the right amount, but then with the noise you're actually creating a band, so even if you're moving around, you always have something that fits, and as you can see, there's a little bit of broadening of the line, but it certainly gets deeper. So noise, the role of noise, I think, is something that is a bit of an area that hasn't been studied all that much and could yield to surprising results. Now, one interest is, of course, Klaus and I suggest that cryptochromes could be a candidate for this magnetoreceptor, and as has been discussed, there may be multiple states where these radical pairs can occur. And I cannot emphasize this too much. We don't even know what the fundamental radical pair is at this point. So there's a lot of people trying to model radical pair systems with great accuracies and all kinds of couplings and so on. But we have to realize if we don't even know what the partner is, there's still fundamental questions to be answered. And they also matter if we want to address questions like the Wilschkus have done with the different colors. Obviously we want to link maybe transient absorption studies or EPR studies on cryptochromes with calculations, but we'd also like to see, are there models that could explain this light dependence, the light effects on the magnetic compass, and so forth. So these are directions that are phenomenological directions that are studied at the moment. And the final direction I will just want to touch on in terms of the theory side is that, what about, this canonical radical pair theory is a bit like the hydrogen model of the radical pair theory. What about extensions and variations thereof? There's one interesting one from Marshall Stoneham from a few years ago where he suggested that the readout may actually not be the biochemical change of concentrations through the radical pair mechanisms, but it may be the effect of such a reaction on transitions in optical states of a nearby visual photoreceptor, and that would provide very different pathways and things to think about. I will not mention, I'm assuming Janos will talk about this. Essentially, there's interest to look at these transitions in and out of these coherent state and see, is there quantum mechanics in these transitions as well, and are there effects, other quantum effects that can matter, and so that I assume will be addressed further. Now, experimentally, and that's where my heart has been for the past number of years. The big question, and the reason we're doing all of this, is we want to find out, is this really the basis of magnetoreception? I mean, quantum mechanics is nice, but it's even more exciting if you can show that it, in fact, underlies a fundamental sense in biology. Now, what has been done is something like this. You have an animal, and you knock out the cryptochromes or knock them down in some form, and then you see, or you have some other organism, where you see that the magnetic field effects are either reduced or go away when you take out the cryptochromes. And that's the first step, but it's not going to be convincing ultimately. And I think what has to be done is that you bring in a second leg, which is that you can do studies at the protein, and you have some way to read out essentially what the magnetic fields do at that point. So to just give, Roswitha was talking about these effects where you just look at magnetic fields in the dark, and we had a fun discussion here on this. Could this be neuronal things? Could this be something else? And that's typically the kind of discussion that you have. Now, if you have a way that you can look at the protein and look at magnetic field effects and have a readout for this, then what you can do, and this is now work, so Roswitha mentioned, Margaret Ahmad has looked, you can get similar responses in plants where you have magnetic fields affecting the growth responses of plants. You can quantify this. This is what Margaret Ahmad, our collaborator, has done in Paris. But then you can go to the protein, and there's a couple of ways how you can read out whether magnetic field effects affect the protein. We've spent quite a few years on looking at a byproduct of cryptochrome activity, which is reactive oxygen species production. But we think a better and more specific readout is to look as phosphorylation of the protein. So the protein, essentially, when it becomes activated, there's an occluded site, the C-terminal, where you have all the coupling partners for signaling, and it becomes open, and so then you can have binding to that. And as part of that, you get phosphorylation of this C-terminus, and so therefore you can look right at the protein, and you see essentially changes in enhanced weight of the protein. And you can then look at magnetic field effects on this. And what you see now is that under darkness, or if you have the magnetic field on all the time, you will get the same effects. You will get magnetic effects, but you will not get magnetic effects if you switch on the magnetic field only during the light phase. So that argues strongly that we're not dealing with some neuronal effect here, but this is something that is linking with the protein. And I think that's the power that you have when you have the protein and the molecular biology in the mix, and not just the behavioral level. However, for this to really work, you also need to have some way to have a high throughput screening, because it is just too slow to do these things. And so Margaret has worked on that and has been successful now through a yeast assay to get, so she started with a library of about 200,000 mutants, and there are other ways to do this, but that's sort of the standard way. And then she identified a number of candidates where you have stronger activity of the cryptochromes. And so one of them here is interesting. It's like by the binding pocket. Interestingly, other mutations around the binding pockets don't do much. And so that's something you can find out at the protein level that you wouldn't be able to necessarily guess or predict otherwise. Now, what you would really love to do is, this was just looking for slightly enhanced activity, but could we get a cryptochrome that really works differently, that maybe is magnetically active under red light or something like that? And if you can show that at the protein level and then put that into some animal and show that now the animal is magnetically orienting in red, I think then you can make a very convincing argument that cryptochrome is really involved in magnetoreception. And what you need, obviously, is also some organism where you can do this, and likely invertebrates are the first candidate for that to look at this. So I just want to pause here for a second. I've been working in this field for now nearly 20 years. But I think the techniques are starting to come together, that maybe not with the next grant, but I mean maybe with the next two. So in the next 5 or 6, 7 years, I think we can really ask the question: is this going to be, is cryptochrome the magnetoreceptor or not? And that's really exciting to me. Another direction that I want to just hit on is we have so many studies where we look at just the cryptochromes and transient absorption and so on. But what happens afterwards? What is actually the signal transduction mechanism? What are the integration, the amplification mechanisms? And what is the minimal thing you need to have an actual readout of the biological readout, not some optical thing, but something that occurs in biology? And I would strongly argue that ideally, a bacterial system would be great to do this because that's where you know everything about signal transduction. And so as a first step, one creates some cryptochrome two-component system. And long term, you would want to have a bacterium going up a magnetic gradient. And if you can show that it's done with cryptochrome, you again have another way to prove that cryptochrome is the magnetoreceptor. So finally, last quick blip. Even if we have shown at some point, hopefully in 5 years or so, that cryptochrome is the magnetoreceptor, it still doesn't prove ultimately that this is based on quantum mechanics. It is making it the case much, much stronger. It's not obvious what else it could be. We had developed this, in some sense, blunt tool of applying radio frequency oscillating fields to try to create noise that destroys the magnetic compass of birds. And it was a fascinating part or time with the Wilschkus together where we really applied these things at birds and saw what happens in orientation, disorientation. We had fun with this. We looked at like one specific frequency. But the problem here is, in a way, that when you apply noise, it could do a lot of things. And it's also, since we don't know exactly what the molecules are that are involved in the radical pair, it's hard to do this fundamental modeling to bring the two together. What in the future I hope we can do, and that again goes to the high throughput, is instead of just creating some sort of noise to disrupt the magnetic compass, is to shield the static field and apply some weird oscillating or quantum control spin echo sequence that is only going to have an effect on the radical pair system, and see that we can recover magnetic orientation. But the problem here is bigger because there are free parameters, like what is the spin correlation time, and you actually have to test this multiple times to get there. You cannot do this with the birds. You can't have a 20-year program to just test the spin relaxation times. But if we have high throughput methods to look at that in cryptochromes, you could conceivably tie down these parameters enough, if you can see that at the protein level, to then try it with birds. And so that would be an exciting future direction, but it's still a little bit in the future. So with this, I'd like to end with this plot and the famous stochastic Liouville equation and the bird having to think about it, that we submitted a number of times to different journal covers, but they haven't liked it yet, so maybe we'll wait in the future. So thank you.
Thank you, Thurston, for a very nice talk. Questions?
Yeah, so maybe you said it, but microphone.
Oh, yes, yeah. Good point.
As far as the in vitro experiments on the protein level, have you seen directionality dependence of the magnetic field?
If you want to do that, so the short answer is on these studies, no. But if you want to do that, you have to either immobilize the protein in some form, or make the spin the tumbling less in some viscous media. There has been one study where you used Peter Hoare lab used a triad of cryptochrome and porphyrin. And they either immobilized it on matrix, or what they did is they just had some viscous material, used polarized light to just have a part of the cryptochromes being activated, and then you could see a compass in it. But it's essentially another step to build this, and so that hasn't been done in most of these experiments.
Thank you, Thurston. Next question.
That was very interesting. I have a couple questions for you. One, these cryptochromes have many other functions like circadian rhythms and all these things. So if you think in terms of evolution, what they should be evolving for both functions, how do you see that? And the second one, even if it's a magnetic receptor like you propose, it has to be coupled to something else. So how that would work on the bacteria, since the bacteria wouldn't have the remaining of the system, basically. So how do you believe the bacteria could now become being able to sense direction unless they have whatever is the other complex machinery the bird has?
Yeah, well, good questions both. So the cryptochrome is, in biology, or the same protein can have multiple functions. In fact, even cryptochromes in the clock. In some organisms, it is part of the clock. In other organisms, it's the photoreceptor that tells the clock that there's light outside. And so I don't think there's any problem with specializing. There's actually 4 different cryptochromes. So it's, yeah, there are different cryptochromes in there. So one could have specialized for that. So I don't see a fundamental issue with that. With the bacteria, the idea is that you actually hijack, let's say the chemical reception mechanism or some other mechanism where, and that's the task, right? You need to build somewhere where you link the cryptochrome and the output of the cryptochrome with, what you get within the bacterial system. So that's what I meant with the 2-component system. You need to have something where you create this link. And once you have that, then you essentially just hijack the whole apparatus, Instead of the tar receptor, you have, the magnetic input into this, and then you go from there.
Thanks, Thurston. One last question. I think somebody... No? Anyone? Okay, another question from there. This will have to be the last one.
Hi, I'm an experimentalist, so I would have a question on the theory side. So why is entanglement necessary for the radical pair mechanism?
So there's a subtle difference between entanglement and coherence that I won't get into here. So it's essentially what the nuclear states do. But coherence is necessary in order to have essentially the evolution of these electron spins. And the fact that you have 2 electron spins on 2 different sides. And so if there's no spin correlation between the 2 of them, what state they're in doesn't matter. It doesn't give you something that goes downstream. And so you need to have that. The way I think about it is actually maybe slightly different. You need to start out with a very ordered system. And it breaks down. At the end of it, it's no longer there. So this is not some, steady state effect. If you look in steady state, there's nothing, right? This is a dynamic effect. This is like you have 1,000 pendulums that you put on their heads. And, in normal random things, 500 would fall one way, 500 the other. But now, because of these slight biases, maybe 550 fall one way and 450 the other. So you've destroyed this ordered state. But in this destruction, you learn something about what was happening or what was influencing you. And I think that's what's happening. So you need this ordered state first. And that's where you need the quantum coherence.
Okay, one last quick question, please.
Just a quick question in terms of biology. It seems to me that it's the lifetime of the diradical. And because the electrostatics of the protein will be different. For example, when you have the trip radical and the flavin semiquinone, you generate a positively charged tryptophan on the surface of the protein. And so electrostatics could play a role in protein-protein interactions. But that may be going on all the time, right, in light. The question is, does the magnetic field then increase the lifetime of the diradical?
So the lifetime is, again, a separate issue, I think. That's much more, dependent on how close they are really and how the electron transfers happen.
So they're far away, of course, in the trip fluid. But has anyone tested, so here's my question.
Okay.
Has anyone tested the impact of the external magnetic field on the lifetime of this separated diradical? Because that could then be integrated into a biological model for what could be happening downstream.
So these scales are on quite different scales. So the lifetimes that have been measured on the times of milliseconds of these radical pairs. The spin relaxation times, you're now like 2-3 orders of magnitude below that, and that's the time where you would affect magnetic effects,
okay.
right?
I think
So the short answer is I don't think it has been measured that directly, but we can have more
We're going to
discussion
have to
on
move
that.
on now. Thank you very much.